So, good evening, dear students. Today we have our resource person is Dr. N.R. Nagaraj saying a few words about Dr. Nagaraj. He is now working as a senior scale scientist in the Division of Crop Improvement at ICR Central Plantation Crop Research Institute, Regional Station Vittal. And Dr. Nagaraj, he had completed his BSc Agriculture from US Bangalore and MSc in Genetics in IARI. And again, he did his doctorate in genetics in IARI. And Dr. Nagaraj has a number of awards and honors to his credit. He is accepted as a PG teacher in the University of Agriculture Science, Bangalore. And he has obtained the Certificate of Appreciation in the bronze category in recognition of the contributions towards building a great nation from the Ministry of Finance, Government of India. And he has a number of membership in professional bodies such as Indian Society of Plant Genetic Resources, New Delhi, Agrobio's newsletter, Rajasthan, etc. And Dr. Nagaraj has a large number of publications to his credit in reputed journals. So, so today I invite you to host this session for the KGRP session. Now it is on to you, sir.
Thank you very much, Dr. Nima, for a nice introduction. A very good evening to all. Hope all are doing good and staying safe. Welcome to today's session on Arecanut improvement and production technology. Just I'll share my presentation.
So we can see your slides.
Yeah, slides are visible. You can see.
Yes,
no?
sir. It is. Yes, sir, it is visible, sir.
Just I did to. Okay. Okay, shall I start now?
Yes, sir. Sir, if it is okay, you can put your video on, sir, so that the students can get acquainted with you.
Sometimes because of the network issue, if I put my video, it may.
Yeah, it is okay. It is okay, sir. Yeah. Okay, sir. Thank you, sir.
Okay, welcome to today's session on Arecanut improvement production technology. I am Dr. Nagaraj N.R., scientist, plant breeding, ICR, Central Plantation Crops Research Institute, Regional Station, Vittal. We take Central Plantation Crops Research Institute. You may be knowing that under Ministry of Agriculture and Farmers Welfare, Government of India, there is an autonomous body functioning. It's called Indian Council of Agriculture Research, under which more than 100 research institutes are there. For all these research institutes, ICAR given some mandate crops. For CPCRI, ICAR given mandate crops like coconut, Arecanut, and cocoa. Our CPCRI's headquarters is at Kasaragod in Kerala state. Under CPCRI Kasaragod, 2 regional stations are functioning. One regional station is at Kayankulam in Kerala state again. One more regional station is at Vittal in Dakshina Kannada district of Karnataka, where I am presently working. CPCRI also has 3 research centers. One research center we are having at Kaikochi state in Assam, Kaikochi in Assam state. One more research center we are having at Mohitnagar in West Bengal. Another research center we are having in Kidu in Karnataka. Along with these regional station and research center, our CPCRI Kasaragod also housing All India Coordinated Research Project on Palms. This ASCP palms spread all across our country in 30 centers. ICR CPCRI also has 2 Krishi Vignana Kendras, 1 KVK we are having at Kasaragod, 1 more we are having at Kayankulam or Alappuzha in Kerala. If you come to ICR CPCRI regional station Vittal, during 1940s, there were so many problems faced by Arecanut industry. And to study this problem, ICAR constituted a committee. That committee studied the problems faced by Arecanut industry and they submitted a report to ICAR. In that report, that committee recommended that to do research on Arecanut, one research center should be established. Like based on that committee's recommendation, Central Arecanut Research Station was established as Vittal in Dakshina Kannada district of Karnataka during 1956. Earlier, during 1956, the CPCRI regional station was established as Central Arecanut Research Station. During 1970, ICAR made CARS as CPCRI regional station Vittal. Like this, the CPCRI regional station came into existence since 1956. We are working on Arecanut and we have developed many technologies in all aspects, in Arecanut improvement aspect, production aspect, protection aspect, processing aspect. In all these divisions, we have developed technologies and successfully transferred to farmers. If you come to Arecanut, you may be knowing Arecanut is one of the important plantation crops of our country. Arecanut is mainly grown in India, Bangladesh, Indonesia, Myanmar, China. In our country, this Arecanut is predominantly cultivated in Karnataka, Kerala, Assam, Meghalaya, West Bengal, Mizoram, etc. And it is also known that Arecanut is having a lot of numerous medicinal and pharmacological properties. This Arecanut play an important, prominent role in the religious, social, cultural, and economic life of our people in our country. If you take present scenario of Arecanut, in our country ranks first in both in area and production in the world. In our country, Arecanut is growing in an area of about 7.31 lakh hectares with production of around 13.52 lakh tons. Among our country, Karnataka is the leading producer of Arecanut. This Karnataka, Kerala, and Assam. These 3 states together, they produce more than 90% total production in our country. This is the area production and productivity of Arecanut in our country. This data is taken from Directorate of Arecanut and Spices Development, Calicut. You see, Karnataka, it is leading in both area and also in production. In Karnataka, you are having around 500,000 hectares Arecanut area with production of around 1,081,000 tons, followed by Kerala. It is having around 96,000 hectares with production of around 92,000 tons, followed by Assam. This is how the Arecanut area production and productivity increased over a period of time in our country. If we come to Arecanut crop diversity, there has been reported that this Areca species, this Areca genera comprises of around 76 species. Among these 76 species, Areca catachu is the only cultivated type. Some of the important species include this Areca catachu, Areca triandra, Areca consina, Areca microcalyx, Areca macrocalyx, Actinolatis calaperia, Norman bia, Norman behi. This Areca palm, which is highly cross-pollinated in nature with chromosome number of 2n is equal to 32. This is how this Areca species distributed all over the world. You can see how geographical distribution of this Areca species, all these different Areca species. ICR CPCRI has the largest collection of Arecanut germplasm in the world. We are having total of around 178 Arecanut germplasm, which comprises of around 155 indigenous collections, which are collected from within the country and also 23 exotic collections. These 23 exotic collections are collected from outside the country. Countries include is Fiji, Mauritius, China, Sri Lanka, Indonesia, Saigon, Singapore, British Solomon Islands, etc. These are some of the important species of Arecanut. You see, this is Areca catachu, this is Areca macrocalyx, this is Areca triandra. You see, this Areca triandra, one more species we are having, Areca cancina. These two are suckering type. As you know, this Areca catachu is having only single stem, monostem, but this Areca triandra and Areca cancina are multi-stem, suckering type. This is another species, Areca macrocalyx. This is another one more species, Arachna scalaperia. This is Norman B. Come to the improvement of Arecanut crop at CPCRI through germplasm collection, conservation, characterization, and evaluation for specific economic trait. We have developed and released 11 Arecanut varieties, including 2 dwarf hybrids. At CPCRI, first variety we have released called Mangala. This particular variety released during 1972. Even though it released nearly 40 to 50 years back, still it is having a lot of demand among farming community. This particular variety developed from germplasm introduced from China. Some of the specific characteristics of this particular variety is that if you take Arecanut variety, generally they will take 4 to 5 years for flowering, but this particular variety, Mangala, it is early flowering. 3 to 4 years, it will start flowering. And another important feature of this particular variety is that it is semi-tall in nature. Since the internodal lengths are less in this particular varieties palms, it is semi-tall in nature and it is recommended for dry kernel or chali processing. This Mangala variety will give average yield of around 2.9 kg chali or dry kernel per palm per year. Again, during 1985, 2 more new varieties we have released. One is Sumangala, another is Srimangala. This particular Sumangala variety again developed and released for chali or dry kernel processing. This particular variety is tall type, but not as tall as local varieties. From this particular variety, we can get dry kernel or chali yield of around 3.28 kg per year. Another variety again, Srimangala, we have released during 1985. Again, it is also recommended for chali or dry kernel processing. It will give average yield of around 3.18 kg dry kernel per palm per year. During 1991, one more variety we have released called Mohit Nagar. This is also a yielding variety, developed and recommended for chali or dry kernel processing. This gives average yield of around 3.67 kg dry kernel per palm per year. Again, during 2005, one more variety we have released called Swarnamangala. Again, this is also recommended for chali or dry kernel processing. It is also a yielding variety. It gives average yield of around 3.9 kg of chali or dry kernel per palm per year. Again, during 2008, one more variety we have developed and released called Kaikuchi. It is also for chali making or chali processing or dry kernel processing. It will give average yield of around 3.7 kg dry kernel or chali per palm per year. Again, during 2014, 2 more new varieties we have released. One is called Mangala, another is called Nalbari. The specialty of this Mangala variety is that this particular variety found to be suitable for both tender nut and chali processing. You may be knowing, if you take this part of Karnataka, this coastal Karnataka, this Dakshin Kannada, Uttarakhand, Udupi districts of Karnataka, here they will go for chali or dry kernel processing. Means around 10 to 12 months, fully ripened nut they will harvest and they will go for sun drying around 1.5 to 2 months. Uniformly they will dry it, then they will de-husk it and they will sell it as a chali or dry kernel. If you go to this Shumaga, Chitradurga, Davanagere, Chikmagalur, these districts of Karnataka, there they will go for tender nut or red nut processing. Means they will harvest 6 to 7 month immature nuts, then they will go for de-husking, then they will go for cooking, boiling. After that, they will give color to that and they will uniformly dry it and they will sell it as tender nut or red nut. This particular variety, Madhuramangala, are found to be suitable for both Charlie or dry kernel processing and also tendernut or red nut processing. If the farmer they are going for Charlie or dry kernel processing, it will give average yield of around 3.54 kg Charlie per palm per year. Otherwise, if farmers if they are going for tendernut or red nut processing, it will give average yield of around 2.95 kg dry tender processed nuts per palm per year. One more variety, Nalbari. Again, we have released this particular variety during 2014. It is also high yielding type. It gives average yield of around 4.15 kg dry kernel or Charlie per palm per year. Again, recently during 2016, one more new variety we have released called Satamanga. The speciality of this particular variety is that it is a very high yielding dual purpose variety, suits for both tendernut processing and Charlie processing, and also semi-tall in nature. Because of this internodal length is less, because of this it is semi-tall in nature, and also early flowering. You are getting feedback from farmer is that farmers field is that some of the farmers are telling us that these particular varieties palms flowering in 2 to 3 years. Some of the farmers are telling within 2 years they got around 90% flowering. Some of the farmers are telling within 2.5 years they got around 50% flowering like that. This is very early flowering variety Satamangala, and also it suits for both tendernut and Charlie processing. If farmers they are going for Charlie or dry kernel processing, it gives average yield of around 3.6 kg of Charlie per palm per year. Otherwise, if the farmers they are going for tendernut red nut processing, from this very particular variety, we can get 3.26 kg of dry tender processed nuts per palm per year. Again, along with these varieties, we have released two dwarf hybrids. Earlier, at CPCRI had one research center at Irehalli in Tumakuru districts of Karnataka near Bengaluru. There, because of natural mutations, one Arecanut tree was dwarf. You see this picture. Arecanut tree was dwarf. But disadvantages was that even though it is dwarf, it was giving very less yield. Per palm per year, you are getting around half kg dry kernel or Charlie. Either by taking half kg of dry kernel per palm per year, it is economical to farmer? No. What we did, we named it as Irrelli dwarf. This natural mutant, we named it as Irrelli dwarf, and we incorporated that particular Irrelli dwarf into our breeding program. We crossed this particular natural mutant Irrelli dwarf with our yielding varieties released from ICR, CPCRI Vittal. You have crossed this Irrelli dwarf with Mangala, Sumangala, Srimangala, Mohitnagar, etc. After so many crosses we have made, two crosses. One first cross is this Irrelli dwarf and Sumangala cross. Hybrid between these two, it retained its dwarfness, and also it was giving around 2.54 kg of dry kernel or Charlie per palm per year. This particular hybrid between Irrelli dwarf and Sumangala, we released in the name of Vittal Arachna Hybrid 1, VTLH1. It gives average yield of around 2.54 kg dry kernel per palm per year. Another combination, this Irrelli dwarf and Mohitnagar, the hybrid between these two, Irrelli dwarf and Mohitnagar, also retained dwarfness, and it was giving around 2.64 kg of dry kernel per palm per year. This particular hybrid is released in the name of Vittal Arachna Hybrid 2. You see, these dwarf hybrids compared to tall varieties, yield is less only. For example, from tall varieties or semi-tall varieties, you can get average yield of around 3 to 4 kg of Charlie per palm per year. But from these dwarf hybrid, you will get around 2.5 kg dry kernel per palm per year. But here advantage is that, for example, you take tall varieties, now crop is ready to harvest, or it is affected by some pest or diseases. To harvest, you have to climb the tree. Again, to spray some pesticides or fungicides, anything, again you have to climb the tree. So here labor charges will be more in case of tall or semi-tall varieties. But in case of dwarf hybrids, the labor charges will be less, because these dwarf hybrid, they'll start flowering, bearing around 1 to 2 feet height from ground level. By this way, you can reduce the cost of production or labor charges in case of dwarf hybrid. Like this, they will be advantages to farming community. Other than this CPCRI released varieties, we are having some of the local varieties also. They are also, these local varieties, they are also performing well in those particular local areas. Some of the local varieties includes the South Kannada local variety. It is famous in Dakshina Kannada district of Karnataka. And Tirthalli, this particular variety famous in Sumangala district. Again, Sagar, again, it is a local variety of Sumangala district. Again, Srivardhan, it is in Maharashtra, famous variety. Again, Sirsi Areca selection one, again, it is famous in Uttarakhand district of Karnataka. These are some of the famous local varieties of Arecanut. Through our improvement program, we have also identified some of the donor parents with the desirable trait in Arecanut. For example, if you want some specific trait like high yield, almost CPCRI released varieties, almost all are giving high yield. And also if you want early bearing, early flowering nature varieties means you can go for Mangala, Satamanga. Like that, for some of the specific traits, we have identified some of the donor parents also. You take this Arecanut or coconut, cocoa, any crop, any perennial crop. Here, top priority, we have to give it to quality planting material. Because you take Arecanut. Arecanut, if farmer, he planted some seedling today means it will take 4 to 5 years for flowering. Again, for yield stabilization, it will take 7 to 8 years. After 7 to 8 years, if farmer comes to know that whatever the seedlings he planted in his garden, it is not of good quality, how many years he will lose? How much money you have invested to raise that particular garden, or how much efforts you would have put to raise that particular seedlings? All these things will go as waste. So because of that, you have to give more importance to quality planting material in case of perennial crops. At ICR CPCRI also, we are producing lakhs and lakhs of quality planting materials, and we are supplying to farmers. At CPCRI, to produce quality planting material in case of Arecanut, we are following 3 stages selection. First stage is selection of mother palms. Then second stage is the selection of seed nuts from these mother palms. Again, after selecting the seed nuts, you'll be sowing in the nursery. Once seedlings are ready to plant in the garden or they are ready to distribute to farmers, again at seedling stage also, by looking into specific characteristics or traits, we are doing selection. Then only we are giving it to farmers. And coming to first stage, selection of mother palms, by seeing specific characteristics or some of the traits, we are selecting mother palms. If you want to select it as a mother palm, it should be attained more than 12 years of age. It should be early bearing type, early flowering type. It should be regular bearing type. It should have more number of leaves, and it should have more than 10 leaves. It should be free from pests and diseases. Like this, based on many characteristics or traits, we are selecting mother palms. Then from these mother palm, fully ripened nuts and weighing more than 35 grams. This type of seed nuts we are selecting. After selecting the seed nuts, we will be sowing in primary nursery. Means on ground itself, you can prepare a raised bed of around 15 centimeter height and 1 to 1.5 meter width and convenient length, using potting mixture of this fertile soil, any organic manures and sand. You can prepare bed, then you can sow the nuts at 5 centimeter distance in vertical position. Then this Arecanut seed nuts will take around 43 to 95 days for germination. Again, after 3 to 4 months, you can shift to secondary nursery. Otherwise, directly you can sow the selected seed nuts in poly bag of 9 inches into 6 inches size poly bag with 9 holes in the poly bag. For that poly bag, you can fill a potting mixture of topsoil, FIM, and sand in the ratio of 7:3:2. After that, you have to mulch it with available waste material like coconut leaf, arecanut leaf, or paddy straw, and daily you have to irrigate it. And again, you have to provide shade to the nursery. Then once seedlings are ready to plant in garden, while distributing to farmers also, you are doing selection at seedling stage. The good seedling is it should be attain 12 to 18 months of age. It should attain 3 feet and above height, and you should have more than 6 number of leaf. Then the girth at collar region, it should be 6 centimeter and above. Like that, based on some specific traits at seedling stage also, you are doing selection. And some of the important phenomena we have observed in Areca is that we have observed polyembryony in case of Arecanut. In case of Arecanut, from single seed, we have got 2, 3, and 4 seedlings from a single seed, you see, because of polyembryony. And also we have identified and characterized this variegated Arecanut also. Generally, normal Arecanut means they will be fully green in color. Here we have identified variegated type where they will be having this yellow and green variegation on leaf sheath and greener portion of the stem, and even the on husk also they will be having these variegations of green and yellow region. Even the kernel color of this variegated type and normal type are different. You see, this is in case of Sumangala local variety, this variegated type. Yes, you can check the kernel. This is normal kernel, and this is variegated type kernel. See, this variegated type. We have characterized this variegated Arecanut for their biochemical triads and physiological triads also. And also, we have documented this chlorophyll deficient seedlings in case of Arecanut, mainly in Satamangala variety of Arecanut. Here you can see these are all normal seedlings, but seedlings we have identified where they lack chlorophyll. We have characterized also these chlorophyll deficit seedlings of Satamangala variety. You can see the frequency of this albinos or chlorophyll deficit seedlings occurrence. You see, during 2019-20, we have sown around 32,430 seed nuts, out of which around 28,415 germinated, of which around 1,587 and 5.58% of the total seedlings, they were albinos or chlorophyll deficit types. And also, we doubted that these albino seedlings may be of applied type. So we wanted to know whether these albinos are different from normal seedling like that. Because of that, by making use of flow cytometry, we have estimated the genome size of both this normal and variegated type. We did not find any difference in applied level. And also we have analyzed this nutrient content of this albino and normal types. And we found that in case of albino, some of the nutrients like nitrogen, phosphorus, potash, magnesium, sulfur, iron, copper, they were 1.5 to 2.5 fold time higher than these normal types. And also we have identified and characterized some of the rare and novel Arecanut palms. Like you see, one of the Arecanut palm we have identified from Udupi district, where this Arecanut tree, on tree itself, the seedlings are forming from bunches. Even in a single bunch, we could able to recover nearly 86 seedlings of different lengths. And also the bunches of this particular tree were having two types of Arecanut. One is somewhat elongated type, one is somewhat round type. Again, we have characterized this elongated type after observing under microscope. Some inflorescence like structure we have seen inside this elongated type of nuts. Some round type of nuts, we could observe small kernel-like structure inside that nut. One more rare tree we have identified where in this tree, inflorescences are forming and inflorescences directly converting into seedling-like structures. See. Another tree we identified where it was having two palms and two crowns, and both the crowns are giving very good yield. This is another tree, it was bearing multiple crowns. And one more type we have identified where this ripe Arecanut with free kernel. If you have seen this Arecanut, ripe Arecanut, the kernel will be very tightly attached to husk. It is very difficult to remove this kernel from husk, but in this particular type, this kernel is free. It is very easy to remove from husk. And also we have estimated this genome size in case of Arecanut using flow cytometry. We were not having any idea about what may be the approximate genome size of Arecanut. So we made use of this flow cytometer available at our institute, and we have estimated approximate genome size of Arecanut. And also we have developed soilless media for raising polybag seedlings of Arecanut. Presently, we are using the soil FOM sand at 7:3:2 ratio as potting mixture for raising polybag seedlings of Arachna. So we wanted to develop soilless media also. So we conducted an experiment, and we found that this vermicompost media found to be more suitable for raising polybag seedlings of Arachna, followed by vermicompost plus kayak with compost media. And also we have developed dust guidelines for Arecanut. Mainly around 32 characteristics we have considered. You see, these are some of the characteristics included in dust guidelines. You can see lot of variation, lot of variability in case of Arecanut, even for morphology, even nut shape, nut color, plant height. Lot of variation, lot of diversity you can observe in case of Arachna. These are some of the dust triads we have developed. And also we are doing the breeding for biotic resistance and tolerance. You may be knowing in high rainfall area, in case of Arachna, fruit rot or mahali caused by Phytophthora medii is one of the biggest problem during rainy season. So we wanted to identify resistance for this particular disease. So what we did, we have screened under laboratory condition the available wild species and related genera, and also some of the Arecanut germplasm. And we found that most of our wild species like Areca triandra, Areca consina, Norman B, Actinitis calaperia, they showed resistance reaction for fruit rot or mahali disease under laboratory condition. So we incorporated these wild species in breeding program. We have made crossing between Areca triandra and Areca catechu, and also reciprocal crosses also we have made. And also we could get some seed nuts also, and from these seed nuts we could raise seedlings. And we wanted to know whether these seedlings are really hybrid seedlings or self-seed progeny of any one of these parents. So we have developed, making use of molecular marker technology, we have developed molecular marker also for testing hybrid purity. And also regularly we are making use of biotechnology approaches, and we are characterizing our germplasm using molecular markers like SSR markers, RPD markers, etc. We are making use of these biotechnological techniques to characterize our germplasms. And also we have developed tissue culture technique in case of Arecanut. Actually, so many explants we have tried, this Arecanut leaf, Arecanut inflorescence, and immature inflorescence. Out of so many explants, the ICR CPCR is succeeded in immature inflorescence. And also through tissue culture technique, you could develop seedlings, and also seedlings planted in the garden. Now they are bearing normal fruit. These tissue culture plants, they are bearing normal fruits. Now, coming to production technology, based on our research, we have found that for Arecanut, good spacing is 9 feet by 9 feet between palm or between rows. Otherwise, if the farmer, if they are going for Arecanut based cropping system, means along with Arecanut, if farmers are planning to grow cocoa, banana, black pepper, any other crops also, multi-cropping also, it is better to go for 11 feet by 11 feet also. Otherwise, for Arecanut, recommended spacing is 2.7 meter by 2.7 meter, means 9 feet by 9 feet between palms and between rows. Planting time, it depends on intensity of rain. Wherever rainfall is more, in those areas, it is better to go for planting during August, September, October. Otherwise, wherever rainfall is less, there they can go for planting during May, June, means along with onset of monsoon. The pit size recommended is around 2 feet by 2 feet, 2 feet. Otherwise, it depends on the soil characteristics. If they open the 2 feet by 2 feet pits, and half of that pit has to be filled with this top fertile soil, sand, and organic manure. Then they can go for planting at the center of the pit. Then to protect these roots and also to save moisture, that area should be mulched with available waste materials, and also shade should be provided to seedlings by using coconut leaf or arecanut leaf. For coming to fertilizer application, for any crop that matter, it is better to go for nutrient management based on soil testing, because agriculture means not only increasing your income, you have to reduce your cost of production also. How we can reduce cost of production means by adopting small technique in your agriculture practices, means by adopting soil testing. If you go for soil testing, if you come to know that what are the nutrient excess in your soil or what are the nutrient deficient in your soil, those nutrients already exist, no need to apply to your garden. Which are all the nutrients they are deficient, them only need to apply. So that by adopting small techniques, you can reduce your cost of production. Another important thing is that through soil testing, we'll come to know the availability of nutrient in your soil. If one nutrient is more, it may make, even though other nutrients are available in the soil, it may make it in such a way that it's not able to absorb by plant. It will create nutrient imbalances. Because of that, based on soil testing only, it is better to apply nutrients to garden. Otherwise, if farmers are not done soil testing, our general recommendation is that on arecanut tree of around 3 years and above age, it requires around 100 gram of nitrogen, 40 gram of phosphorus, and 140 gram of potash per year. Along with this, around 12 kg of organic manures and 12 kg of green leaf manure, or 12 kg and 12 kg of compost or cattle manure has to be applied. Another important thing is that these arecanut roots are spread up to around 75 centimeters from arecanut base. So because of that, it is better to apply nutrient from 1 to 1.5 to 2 feet distance from arecanut stem. And also, at least recommended fertilizer can be divided into 2 splits, and one dose may be given during February, March, April, May time. Another dose can be given during September, October time. And also, regular recommended cultural operation has to be done. Coming to irrigation, there are so many methods of irrigation like hose irrigation, sprinkler irrigation, drip irrigation, flooding, basin irrigation. Through our research, we found that for arecanut, drip irrigation is best. Through drip irrigation, per day, around 16 to 20 liters of water is enough. Excess water also not good for arecanut. And also another advantage is that if you are having drip irrigation, you can go for fertigation. Means application of nutrients through irrigation water. Through this way, through fertigation, you can save fertilizers to the extent of 25 to 50% of the recommended fertilizer dose. And also, wherever rainfall is high, it is advised to give drainage. For arecanut, from seedlings to adult palms, shading is necessary because these arecanut palms are highly sensitive to, susceptible to sun scorching. So shade has to be provided to arecanut palms. at seedling as well as adult palm stage. Coming to cropping system, my question is that growing of Arecanut alone or going for cropping system means along with Arecanut, you can go for multi-cropping. Because of so many reasons, adopting cropping system is beneficial to farmers, because this Arecanut after planting, it will take long period for flowering, and during initial years farmers will be getting very less returns, and also fluctuations in market. Sometimes unexpected losses will be there due to pest and diseases, and also due to natural calamities, and also to efficiently made use of these natural resources like land, sunlight. It is better to go for Arecanut based cropping system instead of monocropping of Arecanut alone. And while going for Arecanut based cropping system, precaution should be taken that the crop which you are including along with Arecanut should not exhaust soil fertility, and also it should give maximum return to the farmer, and also the component crops should withstand shade and heavy dripping during the monsoon, and also the choice of intercrops depends on local need, local market. And you have tried so many medicinal and aromatic crops in Arecanut garden also, and we found that we can grow these medicinal and aromatic crops, and also farmers can increase their income. We have included these medicinal plants like shatavari, vetiver, long pepper, brahmi, etc. And also we have included aromatic plants like lemongrass, palmarosa, basil, davana, etc. These are some of the photographs of Arachna based cropping system. Some of the medicinal and aromatic crops grown in the Arachna gardens. And also some of the flower crops also we have grown in Arecanut gardens. They also performed well. Some of the vegetables also. And also you have done some research on high density multi-species cropping system in case of Arecanut, and we found that in Maidan parts of Karnataka, along with Arecanut, they can grow pepper and cocoa, or along with Arecanut, they can go for banana and acid lime. In coastal Karnataka and Kerala, they can, along with Arecanut, farmers they can go for 3 more crops like cocoa, pepper, banana. In North Bengal region, along with Arecanut, pepper and banana or pepper and acid lime can be grown, and farmers can increase their income. In Wayanad district of Kerala and Uttarakhand district of Karnataka, we have found that farmers they can grow cardamom along with Arecanut. Otherwise, along with Arecanut, they can go for pepper, cocoa, and banana. This is the model of Arecanut based multi-species cropping system. If the farmers, if they are growing Arecanut at a distance of 9 feet between palms and 9 feet between rows, in between four Arecanut palms, they can go for planting cocoa. Cocoa can be planted at 9 feet into 18 feet, otherwise 18 feet into 18 feet. In a row, can go for 9 feet distance between 4 Arecanut palms, and between rows they can go by 18 feet. Then again, in another row between 4 Arecanut palms, they can go for banana. And also from Arecanut base, around 2 feet distance in the north side, they can plant black pepper and it can be climbed onto Arecanut stem. Like this way, in a single area, along with Arecanut, 3 more crops can be grown. This is the methodology we are recommending. Arecanut is spacing 2.7 meter by 2.7 meter, means 9 feet by 9 feet. Then in hectare, around 1,370 population of Arecanut will come. In pepper, again 2.7 into 5.4 meter distance, around 685 pepper will come. Again cocoa 9 feet by 18 feet. In banana also 9 feet by 18 feet. Thank you.
Hello, sir.
Hello.
Thank you, sir. Now the session is open for discussion. Students, you can ask any doubt you have.
Yeah, one question I would like to ask to Mr. Nagaraj, sir.
Yes, sir. Yes, sir.
This is Sunil, sir.
Yes, sir.
I would like to know what would be the best variety for the dry Arecanut for Andhra region.
Chali making.
For Andhra region, yes, dry nut.
Dry karla, chali preparation, no? Yeah.
Yeah, not for cooking purpose. The other stuff.
Chali making.
Dry nut.
Yeah, you see, we have developed and released around 11 Arecanut varieties, including 2 dwarf hybrids. Since this Arecanut, since Andhra Pradesh is a non-traditional area, as of now, none of the recommended varieties are available. But farmers' feedback is that Mohit Nagar is performing well there like that. Am I correct, Sunil?
No, as per my father's experience, what he observed is in a 40-acre farm, this Mohit Nagar is also not giving good yield.
Yeah, I told you, no, since it is a non-traditional area, none of the Arecanut varieties are recommended to Andhra Pradesh. In future, through some AICRP centers, if some research work is done, based on our research only, we can recommend. As of now, none of the Arecanut varieties are recommended to Andhra Pradesh.
Okay. Okay.
Thanks, Sunil. Any other questions?
This is Marina here. I wanted to know what
are
the varieties of flowers that you can grow along with nut, because flowers were during the session, and I wanted to know.
I think your question is not that audible. Can you speak a little more loudly, Marina?
Yes. I observed that there were few varieties of flowers that were grown along with Arecanut. So could you just mention few so that I can make a list of it and keep a note of
it? Hello, Marina madam. Yes, madam. It depends on stage of Arecanut palms also, age of Arecanut palms also. During early stages, madam, you can grow chrysanthemum, gladiolus, all these things.
Okay.
If you want more in-depth information, free publications are available in our website also. It can be freely downloaded from our website. Otherwise, we are having our ICR CPCRI Institute mobile app also, Android mobile app also, eKalpa. It can be downloaded from Google Play Store. Otherwise, you can personally also contact us. We will guide you.
Okay, sir. Thank you so much.
Okay, ma'am.
One more question, Nagaraj sir.
Yes, sir.
Sir, what would be the best size of the seed for the germination purpose?
Yes, sir. As I told you, sir, if seed nuts fully ripened one and weighing more than 35 grams is good, sir.
More than 35 grams. Okay.
More than. Even
Okay.
we are getting seed net, some seed nuts are weighing around 70 gram, 80 gram, 90 gram like that also.
Okay.
It
Okay.
should be
Thank you.
fully ripened one, and
Okay.
selected from mother palms. Whatever
Yeah.
the characteristics are traits I described to call it as a mother palms.
Okay.
Yeah, it should be more than 35 grams, sir.
Okay. Okay, one more thing. What would be the best way to get the germination? Is it in the raised bed, or if you put, or it is suggested to put the seed nuts directly into the packets? What would be the best?
Yes,
Where,
sir.
where do we get the best rate of germination here?
Yes, sir. Better you can go for primary nursery. First, after selecting the seed nuts, you can put in raised beds.
Raised
Because
beds.
earlier
Okay.
you used to do primary nursery, then only you used to shift to secondary nursery. But nowadays, everywhere farmers' complaint is that labor problem. Everywhere labor problem. Because of that, directly we are sowing in poly bag. That technique also we have standardized. Otherwise, first after selecting the seed nuts, as early as possible, immediately,
Okay.
as early as possible, you can sow in raised beds of around 15 centimeter height.
Okay.
Around 1.5 meter width and convenient length. Okay, then you can sow the
Okay.
selected seed nuts at 5 centimeter, 5 to 10 centimeter distance.
Okay.
3 to 5 months, you can transfer to secondary nursery. The secondary
Okay.
nursery also you can in raised bed also okay. Otherwise, poly bag directly you can transfer. Nowadays,
Okay.
in vermicompost media also we are getting very good Arecanut seedlings. Otherwise,
Okay.
potting mixture you can use a fertile soil, sand, or FOM in 7:2:3 ratio.
Okay. Sir, will you be able to share us this presentation via email?
And definitely, sir, it will be there in our YouTube channel, sir. Anytime you can watch.
Sunil, it is live streamed on YouTube. So you can actually, along with the flyer, I have given the link to get the YouTube video. And again, we will be giving an e-manual with the notes. And with all this YouTube videos, we are planning to make a CD also. So that will be done. So for the time being, you can, if you want to see this again, it is on the YouTube.
No, it's not about to watch it again. I just wanted to have some the pictures and all. I want this slide deck.
Sorry, Sunil. Actually, this is sort of copyrighted material. I don't think we will be able to share the slides as such. But you can see, you can view it again.
Sunil, it will be there in YouTube permanently. You can watch slides also will be there. Whatever you watch now, it will be there in YouTube. Even I think they will provide a CDs also, no? They told.
Okay. What I am asking is the actual PDF of this slide, the complete slide deck, one after the one.
No, I don't think we will be able to provide like that as such.
Okay. Okay. Fine. Thank you. That's fine.
So we have Sai Krishnan here. He's saying to you, sir, nice presentation. Thank you, Sai.
Okay, thank you.
So any other doubts are there?
Yes, ma'am.
So
once we have identified the mother palm, is it okay to take the fallen nuts, or do we have to take the fresh nuts as such, sir?
No. Better to harvest from bunches itself.
Okay, sir.
Still, if you want still more purity, means what we are doing here is that you may be knowing, no, Arecanut is highly cross-pollinated crop. Okay, for example, if farmer planted Mohit Nagar variety, after few years, if it farmers took that seed nuts from that particular palm and sown and raised seedlings, it may not be true to type 2 parental, because cross-pollination nature. Through our research, we found that this Arecanut pollen travel up to 1.2 kilometer radius. Okay, if you want to maintain purity in 1.2 kilometer radius, other varieties garden should not be there. I think it is difficult because nearby many other varieties gardens will be there. To avoid to maintain purity, we will go for intersay mating. Intersay mating means this one Mohit Nagar palm will be there. Once the inflorescence comes out, before it is getting contaminated by other varieties pollen, we will bag it. Okay. Then we will collect pollen from another Mohit Nagar variety and we manually pollinate it. Through this process, through this inter-se mating, if you are producing seed nuts or seedlings, they will be more pure or more of parental type.
Okay, sir. Then one more question, sir. Sir, will there be an yield reduction in the case of chlorophyll deficient plants?
This is good question. Actually, the seedlings, what we got, this chlorophyll deficient seedlings, they are dying within a few months because they lack chlorophyll. Photosynthesis will not be there, so they are dying. Okay, we are doing some research also, some basic studies also. We are doing how we can revert back them on those chlorophyll deficit to normal types like that. We are doing it we are doing it, but as of presently, these chlorophyll deficit seedlings are there. No, they are not surviving. They are dying within a span of a few months.
Okay.
Studies are going on. Okay.
Okay. Okay, sir. Thank you.
Thank you, Aparna. So, anybody else? Yeah, Marina again, come.
There was the again I'll come back to the intercropping with Arecanut where
Yes, madam. Yes, madam. Yes, madam.
pepper be what do you say planted along with the Arecanut.
Madam, I think your voice is breaking, madam. I am not clearly it is not clearly audible.
Sir, I said pepper vines planted along with the Arecanut. So what should be the distance? You said I just missed that point. What should be the distance between Arecanut palm ground root and what should be the gap maintained between the pepper vine?
Means this to intercrop this pepper in Arecanut, madam.
How to do that?
Yes, I'll tell. I got it, madam. Means as I mentioned, madam, this Arecanut roots will spread up to around 75 centimeter radius from Arecanut base. So you can plant this pepper plant around 2 feet away from it, away from Arecanut base, madam. 2 feet.
Okay.
Yeah,
Thank
yeah,
you.
Yeah.
Hello.
Yes, sir. Yes, sir.
Sir, isn't it a good practice to directly make this Arecanut, make the pepper plant creep along the Arecanut plant?
Yeah, definitely, sir. We have done research on this Arachna-based cropping system for decades, and we found that pepper can be grown in Arecanut. But thing is that means after Arecanut attains more than 8 to 10 years of age only, you can go for planting pepper because to climb the pepper requires support. No, no problem, sir. But farmers,
No, you're saying 2 feet distance must be kept between the pepper and the Arecanut roots. That's what
from Arecanut base, you can plant this. For example, you see this photo. This Arecanut is there, no, around 2 feet distance. You can plant this pepper, and it can ground itself. We can come later once it reach Arecanut stem. It can be climbed onto Arecanut stem.
Okay. 2 feet distance must be maintained.
Yeah.
Okay. So altogether, this is I have another question as well for the marketing purpose. Maybe you can answer, I think. So what would be the best variety in the market for this Arecanut in terms of sales?
Sale, as I told you, no, sir. For example, if you take this part, here market is for Charlie, where our institute is there, Vittal. Okay, this Dakshin Kanda, Uttrakanda, Udupi. Here market is for Charlie. Here farmers are getting nearly 500 to 600 rupees per kg dry kernel. Okay, 500 to 600 rupees based on the quality. If you go to that Malnad, Karnataka, this Chikmagalur, Shumaga, there mainly market is for tender nut or red nut processing. Okay, there per kg they are getting around 400 to 500 rupees dry tender processed nut.
Okay.
No,
So both.
as I told you, all the varieties are good. You see, as a plant breeder, it will take around 15 to 20 years for developing one variety. After you have put 15 to 20 years of your efforts and time, then also directly it will not come to farmer. Under state government and under central government, many committees will be there. Okay, it has to pass through all these committees. Then only it will come to farmer. All are good varieties only. Little bit yield differences will be there.
Okay.
If you want high yielding varieties, means per palm per year. If you want 3.5 kg and more dry kernel, then you can go for Mohit Nagar, Swarnamangala, Mangala, Satamanga, Nalbari, like that. If you want early flowering type, means you can go for Mangala, Satamangala. If you want semi-tall nature, means you can go for Satamangala, Mangala, like that. Depends on your preferences. But all are good varieties, sir. All are good varieties.
Okay. Thank you, sir.
It depends on your maintenance also.
Yeah,
Another thing, farmers mistake will do that. Along with Arecanut, if you grow cocoa, banana, or pepper, for all the crops, recommended package of practices should be followed. Farmers, what mistake they will do? They will apply only nutrients and water to only main crop, Arecanut. Some farmers, I'm telling, every year we will visit hundreds of farmers' field. What we observed in farmers' field, I am telling.
Okay.
Mainly for main crop only they will do. For other crops also, based on soil testing, you have to supply nutrients. Okay. If you properly do it, means definitely it will be good.
Okay. Okay, can you just bring out the slide where you mentioned that what are all the quantities required for good yield? In one of the slide you have mentioned that.
Yes, sir. That only I told you. It is better to go for soil test based nutrient management. Okay, in Arecanut garden, at least once in every 3 years, you have to go for soil testing. Based on soil testing only, you have to apply nutrients. That will be good idea. Soil test based nutrient application. If you are not done soil testing, means then our this is general recommendation. Got it?
Okay.
This 100 gram nitrogen, 40 gram phosphorus, and 140 gram potash. It is blanket recommendation, general recommendation.
Okay.
Along with this, around 12 kg green leaf manure and 12 kg compost or cattle manure. Okay, should be applied. If per palm you are getting more than 2 kg of dry kernel, then you have to double this dose also. So instead of this, it is advised to go for soil test based nutrient application.
Okay. Okay. If we get the soil test reports, will you be able to suggest what kind of proportions are required for a palm?
Definitely, sir. Actually, if you do soil testing, soil testing labs will recommend what are the nutrient excess, what are the nutrient deficit, what has to be applied. Everything they will report. They will give. Okay. Still, if you are facing difficulties, definitely our soil scientist will help you. That we will take care. That we will take care.
Okay. Fine. Thank you. I'm not aware of this soil test at all.
You're from AP, you can contact your Department of Horticulture or Department of Agriculture or Krushi Vignana Kendras. In our country, for every district, one Krushi Vignana Kendra will be there, KVK. There also, they will be having facility. You can contact them.
Okay. Sure. Thank you.
Yes. Any others are having any doubts? No? So shall we wind up the session then? 1 minute.
Yes, Nima, definitely.
Aljo, do you have any doubt, Aljo?
No, ma'am.
Okay. So I just saw your video, so I thought you were going to ask something. So shall we wind up the session now?
Hello.
Yeah, Sunil, do you have anything else to ask?
Yeah. One
please
question,
ask.
sir. Is there any standard size for the packet, I mean, if you put the plants in the poly bag, is there a standard
yes, sir.
size, or we can
Yes,
go
sir.
as per our convenience?
No, I told 25 centimeter and 15 centimeter and 150 gauge thickness, 150 to 250 gauge thickness, and we should have 9 moles. Okay, to drain out
9.
excess water. I told no.
Just can you please get me to that slide? I'll just take a
Yeah.
screenshot.
As I told you, it is available in our website also, and in publications also available in our website in multi-language, in Kannada, English, Hindi. Yeah. Here it is.
Okay. Thank you, sir. That's all.
Sunil, if you are so much interested, you can once visit our Vital station also. You can just write to our director for the permission, and you can go and visit there. I think that will be well and good for you, because I think you are having so many doubts. You are having plantation, right?
Yeah, sure.
What?
I'll do that once. Maybe in the month of December with my father.
Yeah. Okay, fine. Yeah. So anything else is there? Students, how was the exam? Nobody is responding. How was the quiz test? No response. Anit, Anchana, Aparna, Jagjit. No response. I think nobody wrote the exam.
Latest one is extension system, right? The extension quiz. The latest one is that you have asked.
After that, we had the R programming. That test was there.
Oh.
You didn't attend that?
No, ma'am. You haven't sent any.
The WhatsApp you are not looking.
Yes, ma'am. The last message you have sent is innovation in extension delivery mechanism. That is on 19th. And then yesterday you sent one message.
After that, I had sent that.
No, ma'am.
You didn't receive that? Nobody received that?
No, ma'am.
No?
You haven't sent the link of that quiz. The R
I think
programming
before
one.
sending the flyer and the before sending the flyer of Dr. Nagaraj's talk, I think I had sent a link for the no. Okay, then I'll just check it.
Okay, ma'am.
Okay, then I will. 1 minute, I'll just check it. Please wait. Yeah, I have sent, right? Link for tomorrow's exam on R programming. Link will be active from tomorrow 9 a.m. to 9 p.m. I had sent to KGRP group 2, group 1.
G2. Yes. You haven't sent in G2 group.
KGRP you didn't receive it. I think you might have missed it.
No, ma'am. We haven't received it.
Anyway, I'll do one. Maybe I'll arrange one.
Ma'am, your voice is not clear.
Yeah, actually, I had sent to group 1. Yes, I had sent to group 1 only, I think.
Yes, ma'am. We haven't received any.
Yes. I'm sorry for that. I had sent to group 1 only. So I'll do one thing. I'll just tomorrow I'll do the retest again.
Okay.
Okay. Thanks, Jagjit, for informing that.
Welcome, ma'am.
So anything else is there? No? So shall we wind up the session for today?
Yes, ma'am.
Nagaraj, sir?
Yes, Nimaji, you can wind up.
Thank you, sir. So I take this opportunity to thank Dr. Nagaraj for hosting, for being a resource person of the session of KGRP. So the session was very interactive, and I think many people were interested in Arecanut cultivation. And there were so many doubts and all. So thank you, sir. Thank you very much.
Okay, okay.
Okay. I'm going to end this session. Thank you all my students for attending this session. Thank you.